Just before I start, I want to warn you, because I was standing in this corner. I'm going to talk about a topic that I'm probably not the most expert, but I guess I have gained a lot of experience in CIMMIT, so I hope I can fulfill that. So this is just a quick outline. What I'm going to talk about. So just a quick reason why we need crop diversity, and that's also the reason why we need pre-breeding. Then I will quickly explain what it is, pre-breeding itself, and why do we need it. Then I go a little bit into topics that define pre-breeding, such as identifying promising donors, how we do that. Then we need to hybridize, and the development of pre-breeding and validation populations. So crop diversity, we all work in agriculture, and we know that we have probably the biggest challenges ahead since probably history. Genetic uniformity itself increases vulnerability, and we have seen that in the past. For wheat, genetic uniformity also is also leading to reduced genetic gains, and resilience require options, and options require diversity, and that is why I believe crop diversity is actually a requisite for food security. So we have to bring diversity in to be able to make genetic gain and to be able to diversify in regions or countries. So to improve that, so to gain genetic diversity is as a source of genetic variation. We usually define that source as genetic resources, and across the world there are about 7.4 million plant accessions stored in about more than 1,000 or close to 2,000 gene banks. This year you might know that person. This is Tom Payne. It's the head of our gene bank. So CIMMIT holds the biggest gene bank for wheat, with around 140,000 wheat accessions itself. So what is pre-breeding? So pre-breeding refers basically to all activities that relate to to exploring the genetic variation, characterizing accessions, up to using that accessions in crop improvement. So it holds different activities and different aspects. So the Crop Diversity Trust, I'm not sure, it's an organization, they often compare the gene banks with a supermarket. So they say it looks similar. So if you see these gene banks, a lot of boxes everywhere. It's like a supermarket, like a library, or it's maybe like a library. So as a different comparison. And what they think we need to do is basically, it's a supermarket. So imagine a supermarket, and none of that supermarket would have any information on what is in that cans. So they often compare what gene banks have to do is to label basically all the cans in the supermarket. Okay? And that comes to my first topic. So we need to, how do we read that labels, and how do we identify promising donors in a gene bank, so in genetic resources? How do we define out of the 140,000, out of the 7.4 million, what do we use in breeding? And there are different strategies, and I just will name a few. So the first one is probably the most obvious one, that is just ad hoc phenotyping. So we go in the gene bank, we take some boxes, we phenotype, and then we decide if there is something useful. And actually the BGRI, I think, has been one of the drivers of this strategy. They established for UG99, that screening nursery in Nigeria. Probably most of them, or a few of them, have used it. So basically what they facilitated the high throughput screening of lines for stem rust, right? And CIMMIT, I think that was a very successful concept. So they screened, I'm not sure the final numbers, Shrida Bhavani from CIMMIT is leading this. But they have screened thousands of lines for stem rust. So everybody, and it's probably also the first global screening platform itself for such a trait. So CIMMYT liked that concept, and what CIMMYT right now is doing, they want to expand that concept. So we have want to establish a type of global phenotyping network. We call that. So this is based, for example, here's the Rust screening nurseries. Here's the CIMMYT main yield screening nursery. So the idea is to have more of these phenotyping hotspots, basically. So for international network to be able to screen their material there. Okay, this is just another example. This is coming from our physiologist who will talk tomorrow. I'm not sure if he's going to present that photo. He calls that the bottom-up approach, for obvious reasons. So this is another example of ad hoc phenotyping. So they have been within our Seed of Discovery project, or our Masago seed project. They have been genotyping 700 different accessions in the Obregon's fields. So they have, oops, sorry. So what they have done, they have screened them very roughly. Basically, we call that hill plop. So it's just a plot of seed for very quick phenotypic traits. And then they have designed panels of promising lines that they now will use for research. A more focused strategy is often used by our ICADA colleagues. It's called focused identification of germplasm strategy. And what it basically relies on, it relies on or it assumes that certain lines are adapted basically on traits they have been selected or under selection pressure for. So basically they relate a trait-environment relationship to develop subsets and then evaluate these subsets. So just as Ben said this morning, there was actually recently or a decade before a sequencing explosion. So we all know that genotypes sequencing is getting cheaper and we have more and more information. And this also leads now to the idea of using large genotyping or large sequencing for genotyping efforts in gene bank collections. And there are a number of projects initiated worldwide. So our Seeds of Discovery project is one. So I think in wheat they have genotyped up to 50,000 accessions. In maize, I'm not sure, around 30,000. There's a project on rice, cassava, and the Germans, I think they have genotyped now their entire barley population. And the idea is that we narrow the numbers down by doing this kind of what they called core collection. So the genotypic data is basically used to select the most diverse accessions, and then again a smaller collection is built that is then phenotypically evaluated. Okay, and this has also been done in the CIMMYT project, for example. That is an example of Mexican land races. Okay, we want to make this strategy a little bit more focused. So something we have been recently doing is, because we have all these sequencing data, so we thought, okay, maybe we can get more information out of that. And what we are trying to do is trying to impute this sequence data on known genes, so that our gene bank is able to extrapolate what genes actually are in one accessions. So we are looking at 3 different strategies here. Do the sequence data exactly match to the gene and to the polymorphic cause of that gene? Then we look also at sequence data that are linked to that gene, and sequence data that are probably linked to a haplotype. And this is just an example here. So we indeed find some of the GBS tags. They really go into the polymorphic cause or the deletion of certain genes. Okay. So coming with some new opportunities that we are interested in at CIMMYT, or we are not yet applied, but some strategies we think could be useful. And one of them is genomic selection again. So we have heard about it in the morning. So there was a case study, I believe it was in sorghum in 2016 6, so you have the reference to the paper. And what they basically did, they took one of these core collections, so the smaller set of gene bank accessions in their program. They did a cross-validation within that core collection, and then were trying to use these predicted values to apply it to other accessions in the gene bank. And they, well, at least in sorghum, in that paper, it worked very well. So they have a prediction accuracy of 0.8, 0.9, very, very nice. And they think that could be really a way of giving, again, more value to the genetic resources and then trying to select the genetic resources based on the GS prediction. So I think it could have a lot of potential. One other, I think, relatively new opportunity that was presented by some of our German colleagues, Jochen Fritz Longing and Jochen Reif. So they think we can maybe evaluate genetic resources if we put them in a hybrid. So simply, if you take the genetic resource itself, it usually has a poor economic value. So the real value, the real breeding value is often masked. So they want to test if we cross that with an elite line and evaluate the hybrid. So these are the masks that is coming from different bad economic type or genes is basically going away, and by evaluating the best hybrids, so they would use those basically to go further into question. And CIMMYT is interesting in testing those, and they have, I think, already tested thousands of hybrids now in the last few years. Okay, so now that we have defined how we take and what genetic resources we take from the gene bank, so really the next step is how do we integrate that into our breeding pipelines. Just go on. So usually, and most of the breeders might know that, so the best cross is always elite by elite, right? If I cross elite by a resource, usually the economic performance goes down. So we have to get rid of that. And one approach that has been extensively used in CIMMYT is one approach of doing that are intraspecific hybrids. So it's basically a cross between two species. I'm not sure if everybody knows what the synthetic hexaploid is. Yeah.
I investigated synthetic hexaploid roots in Azerbaijan conditions.
Okay. Yes. So you know what it is?
Yes, we got CIMMYT material.
Yeah. Maybe just quickly to explain it. So the synthetic hexaploid is basically a redesign of the wheat evolution. So what we do at CIMMYT, we cross tetraploid wheat or even dicocoides, another wheat species. We cross that with the diploid Tauschiae donor. We go to a F1 hybrid, and we duplicate the chromosomes, and we generate this designed hexaploid. This is usually looking very awful, and we screen for desirable characteristics, get rid of the very awful ones, and cross them then into bred wheat. So it's an elegant, I guess, solution of bringing wild relative into bred wheat. And this has been successful. The problem only is, and our cytogeneticist is working a lot on that, is that the crosses, can we do that more targeted, or can we do it only random? So he's playing a lot in that area. If he does a random cross, usually, so you have to be prepared that everything, whatever can get out of this cross. If he does a targeted cross, what he sees is that the character of the diploid plants is not always expressed in the hexaploid, and there has been literature since a few years that what is genomically going on during the process of developing that synthetic hexaploid. So there are a lot of bits of chromosomes get lost. There's a lot of rearrangement genetically, so we cannot always say, okay, let's say there is a Tauschiae accession that is rust resistant, so not always the synthetic is not always rust resistant. So that's the drawback of that. So he needs to do a lot of crosses. There needs to be a lot of work on the breeder side on really getting rid of the undesirable to make this a success. And some crosses are just incompatible. But I'm not sure if Matthew will show that also tomorrow. There has been really a tremendous, you can see that here, this is one of our international nurseries. So the use of synthetics in our pedigrees has really gone up. So they are heavily used by the breeders. What is going down here is the contribution of each synthetics. So the breeders are still getting rid of the synthetic components, but they maintain a certain amount of the synthetic. And you see here, this is I think lines selected by Matthew, and everything that is you see in orange, these are the best lines in terms of highest biomass in his pre-breeding program. So you see everything that is in orange has a synthetic actually in the pedigree. Everything I believe that is in green has a landrace in its pedigree. But we see that a lot in the CIMID pedigrees, you see more and more synthetics coming up. And they are useful for very different trait diseases, abiotic stresses, drought stress, etc. Okay. Going back again to the genomics, certainly genomics can also help in introgression. And this is just an overview of the strategies used at CIMID and the route we follow. So if we start with a trait, a target trait, so we define usually has that trait a large or is a large effect gene or haplotype known for the trait. If we say yes, we go into marker-assisted backwashing or marker-assisted selection. If we say no, we are looking more at ways such as genomic selection. If there's nothing known, for example, wheat blast is one of our most famous examples right now, we need to go into gene discovery or need to understand first the genetic architecture of the trait itself. And I think Ben said that. I would like to have more opinion of that if we can actually combine both approaches. Okay. So the most famous introgression strategy, I think, is backwashing, and that can be assisted by markers. So it's usually helpful for diseases and pest resistance. So it's usually used to transfer one or more genes. For using the markers for selection, you need to make sure it's essential that you have a close linkage of the marker to your gene or your trait. And you can control the target gene, and I will show you later, you can also control the genetic background. So this is usually done at CIMID, so the breeders do a lot of backwash work. This is just some examples in durum wheat. So these are the different genes that has been introgressed by Karim, our durum wheat breeders in a few years ago. So he used either a classical backwash strategy, so you used to cross the donor with the elite, and you use the marker in your backwash generations, into segregating populations to make sure the target gene is present. But he has also used some different strategy to combine several genes. And the strategy is working well, but the problem that we most of the time have, and that is the point I would like to make, so this is one of the results of the backwashing. This is a translocation of the leaf rust gene LR19 SR24. These two graphs show the yield in a background that is the line Atil C2000. So it's an older line, but a stable line in Karim's program. And you see these two graphs differ, that this is yield in optimal environment, and this is the yield in the drought environment, so stress environment. So in optimum environment, the introgression lines are still okay in terms of real yield, but if we go into the drought environments, the yield dropped significantly, and that is not what a CIMID breeder wants. So we want to have stable, wide adaptation lines, you know. So what he's going to do now, he's going to make another round of backwashing to get rid really of the linkage track. So the message is here, what we often see in marker-assisted backwashing is really linkage track. a nice side effect, however, was that the LR19SF25 translocation has a yellow color gene, PSY1. So the color of these introgression lines was really nice for durum wheat. So they want to have that yellow color.
Yeah, I have a question.
Yeah.
So in your back slide for this F2 enrichment, is it based on phenotyping or is it based on the genotyping?
No, so here we used...
For example, for that SR22 gene,
yeah.
You have to select like the high frequency, you have to improve the frequency in your F2.
Yes.
So is it for 25 and 22 based on the markers?
Yes, it's on the marker.
It's like the ones which have... It's
on the marker. Actually, usually what we do... No, in that case it was on the marker. Yeah. Sometimes what we do in leaf rust, because we can select for leaf rust very well in our Obregon station, so we select first for the phenotype, right, and we take then the resistant lines and genotype the resistant lines. So it's basically on the both, it's on phenotype and genotype. But in this case it's stem rust, so we use only the marker for selection, so increase the frequency of the two rust genes.
I was just thinking, in a way, if we don't have the marker for that gene, so is it possible we can just have that F2 enrichment based on the phenotypic data? For example, we just bring...
Yeah, you can do backwashing without markers, no problem. The addition what the markers give you, it gives you the certainty that your gene is there, right? What you don't necessarily know in a backwash approach. But if you can get a good phenotype, I think that also works. Okay, this is just another example what is introgressed recently in our durum wheat program. So these are just the generations that were grown in Obregon the last cycle. So it's quite a number of genes, but if you look carefully, so basically the main genes are diseases or it's quality, right? So really major genes. And again, these are the elite lines of these of some of these backwash generations, and I just put here the segregation ratios here. So what you see so these are the frequencies basically of the genes in the selected lines. So you see that so usually you would expect a frequency of at least 50%, but most of the time the frequency of the gene, of the desired gene, is less. Okay, this is again coming back that having these genes, most of the time the acronomic type is going, the breeder is nodding here very heavily, the acronomic type is going down, so there is a negative selection, and we have to take care of this. There's two examples here that are around 50%. In that case, I believe the marker didn't work very well. Okay, so we want to speed that up. So going back to speed breeding. So that takes a long time, doing that backwashes in the field, and then maybe, like in Karim's case, starting again because the acronomic type was not good enough, so they are going to backwash again. So this takes, there we are with in CIMID we are quick, we can generate lines in 5 years, but doing that twice or 3 times, so we are in 10, 15 years. And that is just something we need to improve. So I think there are 2 ways you can improve marker-assisted backwashing. So you can do additionally to foreground selection. So additionally that you select for the genes, you can put markers into the background and select as much as possible your recurrent parent. And the second approach is you can control cycle time. So if we can do that in a speed breeding condition, so we can go much faster. One issue why I think marker-assisted background selection is actually not a new approach. So this has been in the literature for years, even 1819s. I think the issue was that is not heavily applied, it's just the genotyping cost. But now we are getting to the genotyping cost where it can get relevant. So there were genotyping costs in the morning, we're talking about $50 for a wheat breeder. This is a no-go. So really what we think for example, the prices, I believe Ravi is talking about for one line to go through a fixed stage is about $5 estimate. So we think we at least need to go to a $5 level to make that successful. And that's where the big issue still is. Okay. But nevertheless, we have tried this. So we have tried a marker-assisted backcrossing approach in a project we have in IWIP, the International Wheat Potential Partnership. So we generate 8 populations using foreground selection first. So the idea is, so there are different strategies in marker-assisted backcrossing. You can put different stages. In this case, we use the 2-stage strategy. So what we basically do, we select for the gene first, the gene what we want, and then we use only those lines and investigate the background of the lines. We call that a 2-stage strategy. So we've done that. So we have used foreground markers. In this case, is flowering time gene markers. We want to develop kind of a type of near isogenic lines. So we have the Intertech guys are here. We have sent that to Intertech. They have a turnaround time of 2 weeks. And then we have added the background on that with 2 different platforms. We used the CAS platform LGZ and resequencing platforms. They have also different turnarounds. So the turnaround time is between 4 and 6, 2 to 6 weeks, 4 weeks, etc., etc. So to do that in total, we need about 6 weeks, right? Lee was talking in the morning. They flower in 4 to 5 weeks. So here's where still the problem is. We have not done that in a speed breeding background yet. So we still need to optimize really the turnaround time. The cost is more or less close. I mean, the LGC is a bit expensive, $13 for, I think it was about 150 markers, but the STAR technology is around $5. So this is kind of promising. So if they can do it in 2 weeks, then we would have maybe 2 and 2 is 4, then we may be able to do it, and that is what we want to test the next cycle now.
In the industries, they are able to do it already, because if you take it RISE...
Because they have their internal genotyping services.
Which can handle probably millions of data points in a day.
Yeah.
So
I think the big issue here in wheat is the complexity of wheat, so we have not managed to have a genotyping platform that can genotype at the same time for the background and the foreground markers, but I think this is coming soon. So if we are there, I think we can easily speed it up. But well, the result of the first back was okay. We selected based on the LGC markers in this case, because they were coming earlier, and we were just testing this new platform with DART. But also at the end, because the DART markers they have a higher frequency, they turned out to be also quite good to recover the background. Okay. So other approaches besides marker-assisted backcrossing, this is an approach that has been used by our Seeds of Discovery project. I believe, I'm not sure if Susanne is still here. No, I don't see him anymore. So he's, I'm not sure if you know Sukhwinder Singh, so he has been doing that. So in that case, they have been creating crosses with one gene bank accession, cross to a lead, and then cross to another elite, a kind of linked top cross population. So and then they used the other elite as another parent in another cross. And they have done an extensive amount of crosses, and I think in total they generated about 8,000 lines or whatever. So it's a massive resource, and as I understand, they are now in the stage of fully evaluating those. So they have sent some of the breeders, and they have sent some lines to different programs for phenotyping. There. Other populations you could generate is, for example, simple real populations. There's also this type of populations, magic populations, so where you again intercross different parents, so it's a multi-parent population, etc. I think which approach to follow really depends on the resources you have and how many accessions you would like to use for how many traits. but it's a massive undertaking, I believe. Okay. Other new opportunities, what we have been thinking about, but unfortunately nobody seems to like it, except maybe those. So this is kind of, can we generate this population again in a speed breeding background, and maybe using genomic selection to drive basically the crossing of this. I won't go to that entire scheme, but the idea is really to intercross, to predict the intercross line, to intercross again, etc., and build up a population that can be phenotyped and is hopefully advanced for that same populations or the traits you are targeting for. So in my opinion, there is much more experimental studies needed, and also simulations really related to that. Development of, so that's my last topic, development of pre-breeding and validation populations. So I've shown you this, panel before. I think this is really a good approach from the phenology program. So they have meanwhile established several panels. They have a bred wheat panel, a synthetic hexaploid panel, a durum wheat panel, a low phenology panel, etc. And they have, they have. I think that the beauty of it at the end is that you can do many things with this panel. You make that effort really once, but they use it really for a lot of research. So it can also foster, I believe, international collaboration and research of cells. So a lot of different groups worldwide work actually with that material. They phenotype together in Oregon, and everybody has its target trait that they investigate. So they do, for example, they do mapping studies with that same panel. And more recently, that's the last example I want to show you, we also use these panels for validation of traits that we don't actually discover in-house. I just want to give you a recent example what we have found. So we have, I'm not sure if now since probably 2012, there has been an explosion really of genes discovered that increase 1000 grain rate. So there was all these different genes, TRSUS, TRGS3, TRGS3R7, so it was, most of them were observed in comparative genetics. And I said, oh wow, that might be a trait that we can look at to improve a little bit the yield component, a strong yield component. So I want to see what frequency they are in our germplasm. So we used one of these panels, and what we found actually was that some of them were not present at all, so these might be candidates to integrase. But the most strange thing really was, and we confirmed that 4,000 times, that some of the alleles swapped. So some of the alleles were not favorable, that were published to be favorable, was completely unfavorable in our germplasm. So first of all, so just having that panel where we can just easily evaluate that gene was actually really nice. And that is really what you, as a second method, you need to evaluate your traits in your own germplasm before you use it. No, I could just have go in and say, okay, let's the breeder select for these genes, but actually would have actually selected against an increase in 1000 grain rate. Okay, so we found some of these genes really doing the opposite than in our germplasm than in other germplasm. And this panel were genotyped, so we simply took the phenotypic data and we quickly did the GWAS analysis on that for 1000 grain rate, and basically found two new very nice QTL in that panel. And instead of going with the published markers, I think we will rather focus on that discovered markers in our own germplasm. And that's about it. So I just want to summarize. So I think all the take-home messages, I believe that pre-breeding is really a time-consuming and laborious progress for all crop breeding programs. I think private sector programs even don't. Most of them do not even have a pre-breeding program, so there's not as much interest, even so it's so important. It requires really long-term commitment and also large field and phenotyping power. I believe that it's important to have a focused strategy. So there are also opinions of say, okay, we do completely unfocused, but I believe it's very difficult, and we have seen that a lot. That some of, sorry, there's an E missing. That some of the core collections, especially identified with genotyping, so you can create the most diverse and highest core collection, but that's probably not the collection you can phenotype. You know, they vary so much in phenology, etc., that it's very difficult. So pre-breeding for simple traits, I think, can be heavily optimized still. So using speed breeding, using the new marker technologies. So pre-breeding for the more complex traits is still difficult, and I sometimes wonder, pre-breeding is not a new thing, right? And integration of quantitative traits is not a big thing. Sometimes I wonder why not more experiments have been done so far. I'm not sure if it's now coming because of the genomics or what is happening there. So I think there's a big need of working on pre-breeding strategies for quantitative traits. So there are much more experimental studies needed. We have to exploit the full genomic toolbox, I believe, simulations, and I believe also international cooperation is there necessary. Okay, thank you.